My name is Nordine Helassa, and I graduated a PhD in biochemistry from the French National Institute for Agronomic Research. I'm a British Heart Foundation Fellow and a tenure track fellow at the University of Liverpool. And my current research interest is the role of calcium binding proteins in cardiac muscle contraction. So why did I choose this particular paper? So I found quite interesting the similarities you can see between cardiac muscle contraction and glucose-stimulated insulin secretion. As you can see in both phenomena, you have a key role of calcium ions and voltage-gated calcium channels in both cases. So in cardiac muscle contraction, when you have an action potential which depolarizes the membrane, then you have the opening of the L-type calcium channel that allow calcium to enter and activate the ryanodine receptor from the sarcoplasmic reticulum, and then you get more calcium into the cytoplasm, which promotes cardiac muscle contraction. So in glucose-stimulated insulin secretion, you still have the same 3 steps of membrane depolarization, activation of calcium channels, and then the physical outputs. The major difference, I would say, is that the way the depolarization of the membrane is created. So in this case, is the entry of glucose, which when it will go through the Krebs cycle, we generate ATP, which will then block the KATP channels. And this is this blockage that will depolarize the membrane and activate the calcium channels. So in their paper, the authors have used quite a nice combination of techniques ranging from calcium imaging, biochemistry, and electrophysiology to identify a new regulator of the calcium, of the voltage-dependent calcium channel, which is Tspan7. So I'm not going to say any more, and I'm just going to hand over to Arya, who is going to describe in more details some of their exciting work.
Right, thank you, Nordine. And firstly, I want to thank the organizers for inviting us as panelists for the Journal of Physiology's Virtual Journal Club. And so for today's discussion, I'll give a brief introduction where I'll discuss the importance of calcium in glucose-stimulated insulin secretion. And I'll briefly talk about how L-type voltage-gated calcium channels, or the CAV channels, mediate calcium entry. And also I'll briefly mention the role of auxiliary subunits in the regulation of CAV channel function. And then we'll dive into what is currently known about tetraspanins in the regulation of CAV channel activity. And this will be followed by a discussion of the results of our study, which will be presented by Matt, who will show you that Tspan7, which is shown here in red, interacts with the L-type CAV channel. And by doing so, it decreases glucose-stimulated calcium influx and insulin secretion. And so a lot of work has gone into establishing the consensus model for glucose-stimulated insulin secretion, which shows that calcium entry through the voltage-dependent calcium channels is critical for insulin secretion. And this was first shown by Gerald Grotsky in 1966, where upon stimulation with glucose, islets that were perfused with medium containing calcium showed the typical glucose-stimulated insulin secretory response. Whereas upon removal of calcium from the perfusion medium, which is shown here in dashed lines, you can see there was a complete inhibition of glucose-stimulated insulin secretion. And so going back to the consensus model, you can see the beta cell here responds to glucose stimulation by increasing the ATP to ADP ratio, thereby resulting in the closure of KATP channels, leading to depolarization of the beta cell membrane. And this activates the voltage-dependent calcium channels, thereby leading to calcium influx and subsequently insulin secretion. And so this beta cell calcium entry upon glucose-mediated depolarization is shown here on the right, where you see an increase in the inward currents to the L-type CAV channels at depolarized membrane potentials. And this inward currents contribute to the upward stroke of the action potentials. And so this action potential firing is in part controlled by the opening and closing of CAV channels that leads to the development of calcium oscillations and pulsatile insulin secretion. And so interestingly, the activity of the CAV channel is controlled by a variety of auxiliary protein subunits. So as you can see here, the alpha subunit, which is the pore-forming subunit of the CAV channel, forms complexes with other subunits like the extracellular alpha-2 subunit, the transmembrane delta subunit, the intracellular beta subunit, and importantly, the 4-pass transmembrane gamma subunit. And what is also interesting is that the activity of CAV channels is also controlled by the tissue-specific expression of these auxiliary proteins, leading to differential regulation of the CAV channel function depending on the expression levels of these subunits in various tissues in the body. And so coming to tetraspanins, I would like to point out that tetraspanins structurally resemble to the gamma subunit of the CAV channels, which was shown on the previous slide. And so as you can see, T-spans have 4 transmembrane domains, a small and a large extracellular loop. And the large extracellular loop has this CCG conserved motive, which is important for protein function. And also it's important for regulation of the interaction of T-spans with its interacting partners. And so a few years ago, a German group showed that tetraspanin 13, which is shown here in yellow, interacts with the voltage sensing domain of the N-type CAV channels and affects calcium CAV channel function in neurons. And specifically, tetraspanin 13 accelerates activation and inactivation kinetics of the N-type CAV channels. And by doing so, it decreases the voltage sensitivity and channel open probability, which is shown here in the black solid line in neurons overexpressing T-span 13. And so what this paper demonstrated was that T-span 13 serves as an important interaction partner of the N-type CAV channels and is likely to play an important role in neurotransmitter release in neurons. And so coming to T-span 7, which is the focus of today's discussion. So T-span 7 is a highly expressed protein in the endocrine cell types, including the alpha cells, the beta cells, and the delta cells in both mouse and human pancreas. And at the protein level, you can see T-span 7 protein is enriched in the pancreas as compared to the other tissues. So interestingly, T-span 7 forms protein complexes with a variety of critical proteins that are involved in various intracellular processes, including the 3 that are shown here. And so T-span 7 regulates F-actin dynamics by interacting with PIC1, which indirectly regulates the activity of the actin-related protein ARP2 and 3, which normally function to reduce cranial exocytosis and hormone secretion. And so then the loss of T-span 7 would be predicted to enhance cranial trafficking and islet hormone secretion at basal glucose conditions. Secondly, T-span 7 also regulates the surface expression of the inotropic receptors that are the AMPA receptors in hippocampal neurons. And as these receptors are also expressed in the islets, you would assume that loss of T-span 7 would increase the surface localization of calcium permeable AMPA receptors, thereby leading to enhanced glutamate-mediated calcium influx and islet hormone secretion. And the last and the most interesting role of T-span 7 in calcium handling is its interaction with the phosphatidylinositol 4 kinase or PI4 kinase, which is important in the regulation of the membrane phospholipid PIP2 expression. And so PIP2 is a known ion channel regulator, whereby increase in PIP2 levels increases KDP channel activity, leading to membrane hyperpolarization and inhibition of voltage-dependent calcium channels. And so it is evident that T-span 7 is likely to play a very important role in calcium handling in the islets. And so based on the information I've told you so far, we asked the question if T-span 7 regulates beta cell calcium handling and insulin secretion. And so this is me at the end of my part here, and I'll hand it over to Matt, who will explain the results of our study.
Hey, so thank you so much, Arya. And also, I would like to take this opportunity to thank our hosts for inviting us here to the virtual journal club today to present some of our findings in this recent paper. Okay, so this is the first figure from the paper. And so this shows some immunofluorescent staining of mouse and pancreas sections that we did, showing high expression of T-span 7, which you can see in green in both beta cells, indicated by insulin staining in red, as well as glucagon staining in alpha cells shown in violet here. And so you can appreciate that within the endocrine islets, it's very highly expressed, However, it is not present as a protein anyway in the surrounding acinar tissue. Also, you can see at the bottom here some more high resolution staining results showing that T-span7 is not only restricted to the plasma membrane of these beta cells, but also it's distributed throughout the intercellular space. And so, and we also observe a very punctate staining pattern. So this seems to suggest that it could serve additional functions intracellularly in the islet in addition to at the plasma membrane. Figure 2 here shows the development of a lentiviral-based shRNA delivery system that we utilize to knock down T-span7 in both mouse and human islet cells. And so just for brevity's sake, throughout the presentation, I'll refer to as knockdown as KD in the text. And so in the middle, you can see this knockdown verified by immunofluorescent imaging. And on the right, you can see this replicated via Western blotting. And so you can see that we got efficient knockdown of T-span7 both in mouse as well as human islet cells. And so throughout the presentation, transduced cells were identified by the co-expression of an M-Cherry fluorescent reporter. In figure 3, I've broken this down into 2 different slides. The first slide here shows the influence of T-span7 knockdown on the glucose-stimulated calcium entry into mouse beta cells within whole islets. And so here at the top, you can see the response to glucose, both at 7 as well as 14 millimolar glucose into these mouse islets. And so you can appreciate that T-span7 knockdown increased glucose-stimulated calcium influx at both glucose concentrations. However, the effect was much larger at 14 millimolar glucose. And then on the bottom, you can see that also accelerated the calcium oscillation frequency of these mouse islets. And so this could be important because as you may all well know, insulin secretion in the body is pulsatile in nature. And so it could be that this is influencing that pulsatile release of insulin. The second half of the figure here shows the effect of T-span7 knockdown on human beta cell calcium influx in response to glucose as well as depolarization with KCl. And so much like we saw for the mouse beta cells, in human beta cells, T-span7 knockdown increased the glucose-stimulated calcium influx. And we also, when we activated KTP and depolarized with KCl, we saw a similar effect. And so this is important because it strongly suggests that T-span7 is having a direct effect on beta cell calcium channels. And so to interrogate this potential effect, we next measured the effect of T-span7 knockdown on calcium currents in both mouse and human beta cells. And so we focused on, for this paper, the effect on L-type calcium channels, as these are the primary channels by which calcium enters into beta cells. And so the way we did this using electrophysiology was we stimulated these currents via the polarization of the beta cell plasma membrane at a variety of different membrane potentials. And then we blocked L-type channels with isradipine and then subtracted the remaining currents from the total currents to arrive at L-type currents. And here you can see the results of the study. So on the top, you can see the effects on L-type channels in mouse and human beta cells. And so you can appreciate that in both cases, we saw a significant increase in the size of L-type currents when T-span7 was knocked down, whereas on the bottom, we saw no effect on non-L-type channel currents. Next, we were interested to determine if this was via a direct effect on the channel or through some sort of indirect mechanism. And so we pulled down T-span7 along with Cav1.2 and 1.3, which are the predominant L-type channels expressed in beta cells. And so in both cases, we found that T-span7 co-immunoprecipitates with these L-type channels from primary mouse or primary human islets. We confirmed this in a heterologous system where we overexpressed T-span7 with HA tagged Cav1.2 or 1.3 and found that once again T-span7 co-immunoprecipitates in the presence of an anti-HA antibody, but not with an isotype IgG control. And so we also were interested in determining if T-span7 affects the surface localization of these channels, but in a heterologous system, we saw no effect, as you can see here in the bottom left. And then finally, we wanted to determine if we wanted to show that we have functional channels. And so we looked at calcium influx through both 1.2 and 1.3 in response to a high concentration of KCl and found that as with primary cells, T-span7, in this case, overexpression led to a decrease in CAV channel currents or calcium influx. And so here in this figure, this is the first half of the figure. This is an electrophysiological analysis of specifically CAV 1.2 and CAV 1.3. And so as predicted, when we overexpressed T-span7, we saw a decrease in the size of both 1.2 and 1.3 currents, as you can see on the top. And we also saw an effect on activation and inactivation kinetics of the two channels in a channel-dependent fashion. And so on the left, you can see that T-span7 reduced CAV 1.2 activation, the activation rate of CAV 1.2, whereas it decreased the 1.3 inactivation rate. And so we did have this sort of channel dependent effect on the kinetics. And here in the second half of the figure, we examine the effect of T-span7 on voltage-dependent inactivation of these channels, specifically recovery of the channels from voltage-dependent inactivation. So basically what we were interested in looking at is that if you expose these channels to a prolonged activation, they slowly lose that activation over time. And so from our results, we see that T-span7 overexpression actually accelerated recovery of 1.2 from this inactivation event, whereas 1.3 was not influenced. And then finally, we wanted to look at another important ion channel in beta cells, which is KATP or ATP sensitive potassium channels. And so in this case, we saw no effect on this channel. And so it seems to be that predominantly that T-span7 is affecting calcium handling in these beta cells through its effect on calcium channels. And so here in the last figure, we examined the functional effect of T-span7 knockdown on insulin secretion from human beta cells. And so these studies were conducted in dispersed human beta cells transduced with either shRNA specific for T-span7 or a scrambled shRNA. And so we looked at both basal insulin secretion on the left, as well as stimulated secretion on the right. And in both cases, we observed a significant increase in insulin secretion when T-span7 was knocked down. And so I just wanted to point out this effect at 5.6 millimolar glucose, which is a little surprising, but not incredibly unexpected, because we did see a slight increase in glucose-stimulated calcium influx under 7 millimolar glucose conditions. And so it could be that we're seeing a shift in the channels to being activated at lower glucose concentrations in the absence of T-span7. And so just to wrap up and summarize our major findings, we find that T-span7 interacts with CAV 1.2, reducing channel conductance and slowing channel activation. We also find that T-span7 interacts with CAV 1.3, which decreases channel conductance and decelerates inactivation kinetics. And these effects together limit glucose-stimulated calcium influx, slow calcium oscillation frequency, and decrease insulin secretion. And so we conclude that T-span7 functions as a CAV channel regulatory subunit in beta cells. And so with that, I would just like to acknowledge the Jacobson lab, as you can see here at the bottom, as well as our collaborators at Vanderbilt, Maria Passafaro, for providing the T-span7 plasmid that was used in these studies, as well as our funding sources. And with that, I would like to take any questions.
Okay, thank you, Matt, Arya and Matt. I think it was a very nice and clear presentation. So I just want to remind everyone that they can use the Q&A function at the bottom of the screen to write any question they have about this presentation and this work. I'm going to give you a little bit of time by starting the discussion. So I had a question for Pei-Ying first. So Pei-Ying, as a senior editor for the journal, can you please tell us what you think made this paper stand out, and also what advice would you actually give to submit papers to the standard of the journal?
Well, I think, thank you, Nordine, for inviting me to this forum. And I think that the thing that stood out about this paper for me is that it's conceptually innovative. Although this phenomenon of the T-span, I think it was 13, being shown previously in a neuronal system, it was taking a concept and extending it to another system in a highly innovative and creative way. So as you know, the Journal of Physiology is quite interested in studies that are non-incremental, but rather are conceptually innovative. And you outlined it also very well when you highlighted the reason that you picked this out for a journal club topic as well. So really, the journal is seeking studies that do detail these sorts of conceptual innovations as well as technical innovations. And in that latter case, that would basically go to a techniques for physiology sort of manuscript. But what does conceptually innovative mean for most people? It means that it drives the field. It drives the field in a new direction, and it'll exert a sustained and powerful influence on the field and possibly even beyond. I sort of saw that when it came into me through the submission funnel through our editorial office. And as a senior editor, I thought that this would be a great exercise as well for a reviewing editor who at the time was an editorial fellow. We do have this program, and so Pavel drove this ship, and he basically sent it out to those individuals who he thought were the most highly qualified to assess his manuscript. The other thing is the technical rigor. The fact that you use many different sorts of outcome measures for probing your question, right? That you simply are not looking at something and putting it into a cookie cutter, and it's not sort of a, well, X does Y, and so we'll see whether X prime does the same thing to Y, right? It is truly a novel and innovative study in my book. So thank you, Nordine.
Yeah, thank you very much, Bing. Yeah, I completely agree about the fact that the use of like multiple different techniques to show the same effects is actually make the paper very strong.
Yeah.
And I think that after reading the paper or listening to the presentation, we can't really argue that T-span is not regulating calcium channels. It's kind of obvious now. So feel free to ask any questions using the Q&A function at the bottom of the screen. I'm going to start asking a couple of questions to the panel to give you a bit more time to think about what you would like to hear about a bit more. So my question is about the human primary cells you have been using. So I can see from the table you provided, the donor tables, that some of them were coming from males, some of them from females, the age groups were a bit different. I was wondering if you saw any gender effects or age effects in how T-span is regulating the channels?
Yeah, that's a great question. So we would love to do these sort of studies, but it's kind of random who we get, right? And so we don't have a huge data set. I will make a couple of kind of general comments though. We do see that above sort of like 30, 32 BMI, sometimes the islets don't perform the same way as BMIs below that. And I know that other people in our lab seem to think that if the donor is a little younger, those islets tend to perform a little differently. I don't know, you can add to that if you would like, David.
Yeah, so the calcium handling is greatly influenced by the age of the donor. And as you age, there's some groups that say insulin secretion goes up, but there's a few groups now that believe that insulin secretion goes down as you age, and especially with environmental context of obesity and excess nutrition. And so this calcium handling defect, we don't know if it's related to T-span 7, but T-span 7 doesn't change dramatically over the age of an islet. And so if it is through T-span 7, there are other modifications on T-span 7 which could be affecting calcium channel activity, but likely there's also contributions of KATP channel activity because mitochondrial function decreases with obesity. And so there's probably other contributions that are affecting calcium channels. There are a few questions now. Do you want me to answer those, or do you want to answer those, Matt? Can you see those?
So it's there is a question from Muhajid Nasir Hussain. So he's asking, can the excess of calcium influx via the membrane affect the rate of insulin secretion, especially to those suffering from diabetes?
So the question is, can the influx affect the rate
of insulin secretion, especially for people suffering from diabetes?
So I'm not sure about the rate, but the total amount would certainly be affected. And I would assume that the dynamics would be as well. I can't say for sure because we haven't done those assays, but I would think yes.
And I can follow up a little bit. So Sebastian Barg has found that in order to get typical insulin granule release, what you need is these calcium channels coming together. You can watch them in real time. They come together and form these clusters of about 8 calcium channels next to each other that allow for proper calcium release at the granule, calcium entry at the granule. And this allows for insulin granule fusion with the membrane and subsequent insulin release. And if you look at type 2 diabetic islets, you lose this clustering, and thus you're losing that little microdomain of calcium. So it's the microdomains of calcium that we think are more important during insulin secretion and are disrupted in diabetic settings.
Thank you, Matt and David. There is a follow-up question from Mujahid, which we kind of already touched upon. So he's asking, is there any differences between the rate of conduction of calcium into the membrane based on body mass index of individual?
So I can answer a little bit. So what we do know is that there's different phases of calcium influx. And so when you stimulate with glucose, what you get is this big burst in calcium influx, which leads to first phase insulin secretion. And you lose a lot of this. And so it's the amount of calcium coming in, and that has to do with rate because you just don't have it coming in as fast. So not as many of the calcium channels are opening, they're not clustering, and you're not getting as robust of a calcium influx in patients with type 2 diabetes. And this is reducing what we know is first phase insulin secretion. It also affects second phase a little bit, but predominantly first phase. And interestingly, different calcium channels affect different phases of insulin secretion. If you knock out Cav1.2 in a mouse beta cell, you lose first phase insulin secretion. Interestingly, if you knock out 1.3, you get compensation by overexpression of 1.3, 1.2. And so this compensates for the loss of 1.3. And so there are differences in terms of these calcium channels and what types of insulin secretion they regulate. And as you talked about earlier with skeletal muscle, some people also believe that the ryanodine receptor is important here and that different calcium channels interact closely with the ryanodine receptor, 1.2 in particular. And this allows for calcium-induced calcium release from the ER, which stimulates more insulin secretion. And that's also disrupted in type 2 diabetes. And Andrew Marks has some nice work on ryanodine receptor mutations that affect insulin secretion and calcium-induced calcium release.
Okay, great. Thank you very much. I have a bit of a technical question. It's, when you do the pull down experiments, so this is what you use to show that T-span is directly interacting with Cav1.2 and Cav1.3. How can you rule out that it's not an indirect interaction, that there is always an accessory protein that is co-precipitated that actually does, the interaction, if you see what I mean? So how can you be sure this is direct?
Yeah, so that's a really great question. And it could be that this is a protein complex. The way we try to minimize non-specific interactions, at least, is we use detergents and BSA and different serums. But yeah, I would fully expect that this is probably a multi-protein complex because we know that T-span 7 interacts with a variety of different proteins, AMP receptors, dopamine receptors, PIC1. So that's actually something we're really currently looking into. We're developing a system where we can pull down T-span 7 from primary cells or even cell lines and send that off for analysis. And we're really interested in building that interactome of T-span 7 for not only beta cells, but alpha cells and delta cells.
So I might add something about that. Actually, when you say you're sending those out for analysis, are you suggesting you're doing mass spec on this to see what other things are coming down?
Yeah, exactly. Yeah.
So another suggestion to get around that would be to see whether there actually is a direct interaction would be to perform some surface plasmon resonance work with synthetic constructs, right? Or pieces of the T-span and then flowing over pieces of this calcium channel and seeing whether or not the binding affinities are in alignment with that kind of interaction.
That's actually a great idea.
That's a great idea, and that's something that we can do. We have now members of the MPB community at Vanderbilt that we've got a new Titan Krios that we can start doing maybe crystallization as well to see if it interacts with the voltage sensing domain. And so when we make the protein, we can try both approaches.
Yeah. And you guys got great toys over there.
It's not, well, they just got it up and running, so we're not going to be the first in line, but we're definitely thinking about those experiments, and that's a wonderful idea. Thank you.
Yep. Looking forward to seeing it.
I guess the fact that both of them are membrane proteins doesn't really make it especially easy, but what's not easy makes it even more interesting. I wanted to ask you a quick question about VDI. So you looked at VDI for these channels and you saw an effect for one of them. Is there, so I'm not very familiar with islet cells, to be honest, but do you have also CDI in this, in the glucose-stimulated insulin secretion? Would the calcium also inactivate the channel?
Yes, it can, and that's definitely something that we in the future should probably look into. We just are kind of working through everything one by one, one step at a time.
Yeah, cool.
But yeah, that's something also for we're interested in the effects on AMP receptors, and so that's also going to be important there.
Okay, great. So there is quite a few questions now on the Q&A, so I'm going to go through them. Someone from the audience is asking, why does T-span knockdown cause a greater reduction in glucose-stimulated calcium entry compared to KCl-stimulated calcium entry?
Yeah, that's a great question as well. This is something that we've noticed. We see larger effects of the knockdown on the glucose-stimulated insulin or calcium entry, and so this is consistent with what we saw with basal insulin secretion. So it does appear that that T-span 7 is important for setting that threshold for channel activation. And while we don't have any proof of anything right now, one thing I've been curious about recently is that I found in a paper that T-span 7 interacts with cellular prion protein, which has been shown to affect iron trafficking in islets and also through that glucose transporters. And so it could be that T-span 7 is playing a role in glucose transport into the beta cells. And so this is something that I'm interested in probing in the future.
Thank you. There is a question from Nicholas Heron. So he says, thank you both for your presentations, very clear. Do you have any evidence that T-span 7 is altered in vivo in diabetic humans versus controls? Is it known how non-pharmacologic intervention affect T-span 7 levels in vivo in humans? For example, the effect of exercise and or diet?
Thanks for the question. This is something I'm very curious about as well. I don't have great answers yet. I can tell you what we know. At least at an RNA level, it doesn't appear that diabetic conditions or stressful conditions affect T-span 7 mRNA levels. There are some studies, though, in the literature showing that in a mouse model, T-span 7 is affected under certain high-fat diet conditions. And so we don't really know how that applies in vivo for humans. It's something that we're looking into in the future. As far as different inputs that could change the levels of T-span 7 in vivo, that's also a really intriguing question that we would very much like to look into in the future. But right now, I can't really comment on that. But it is something that I would like to look into in the future.
Okay, thank you very much. I have a quick question about the Cav1.2 and Cav1.3. So T-span is regulating both, but differently. So my question is, so what do you think in terms of physiology this is actually doing? The fact that two channels that are pretty much doing, the same thing about, letting the calcium in, they are not going to be regulated the same way. And my follow-up question is, in one of your figure, when you look at the IV curve for the Cav1.3, you have a right shift, like the activation curve, instead of being activated at like -20, it's more like close to 0. Do you think this has any significance?
Was there a difference? I thought I analyzed this data and I didn't see a statistical difference
Okay.
on the
I
activation.
understand
I
it.
could have been close, maybe.
Yeah.
Um,
Visually, it looks like there is a difference, but maybe it's not
are you
statistical.
talking about and when we overexpressed it in the hex cells, maybe?
Yeah. Yeah, that's right.
Yeah, maybe I didn't analyze that. It might have just been the primary cells that I analyzed. So it could be that this plays back into what we noticed with the calcium, right? Because if it activates more at more hyperpolarized membrane potentials, it could be that could account for that increased calcium and increased insulin secretion that we observed. As far as, well, can you remind me of the first part of your question? I wasn't sure what the question was exactly.
So one part of the question was, so you have CAV 1.2 and 1.3 that are apparently regulated differently by T-span. So what do you think, what does it mean in terms of physiology? Because so one of them is affecting the activation, the other one is more affecting the inactivation. So what would you say would be the role? Is there like a particular role of these channels?
Yeah, so it seems to be that like the overarching effect is to sort of limit the conductance through these channels. And so it could be to just control, kind of put an upper limit on the amount of calcium that enters into these cells. So because if you have too much calcium over time, that can lead to major changes in the nature of that cell as far as protein expression and transcripts go. And it feels like it's kind of ubiquitous across a number of different calcium channels, right? Because in addition to 1.2 and 1.3 there is 1.2 and 1.3, there is some evidence that it also affects CAV 2.1 in a similar fashion, which is also important in the islet in terms of glucagon secretion from alpha cells. And so it feels like sort of this master control switch for calcium channels.
Okay, thank
The
you.
voltage dependent inactivation is really important as well, because as you build up more inactivation, you're not going to have those microdomain, those little bursts of calcium happening to enough extent to stimulate insulin secretion. So it could drastically affect insulin secretion if you have more inactivation. And interestingly, under glucose stimulation, you depolarize the membrane potential right to the level, the threshold for activation of the voltage dependent calcium channels. And this is controlled by potassium channels, obviously, but as you become diabetic, that changes the membrane potential from where the plateau potential sits. And this really drastically affects calcium entry and would be even further regulated or dysregulated without T-span 7, because you would have more inactivation of those CAV 1.2 channels. And so we're not sure if you want to take it away or keep it. It might be serving an important role in preventing inactivation during sustained glucose stimulation, such as under diabetic conditions.
Yeah, it's a great point. And these proteins, they are important for forming microdomains, these tetraspanin-rich microdomains. And so this could play an important role in that clustering of calcium channels, beta cells. Yeah. Really intriguing concept that we are starting to get into.
Great. Thank you, Matt and David. There is another question from Mujahid. And I will have a follow-up on this one. So he's asking, are there some drugs that affect the activity of T-span 7 in humans? And I also wanted to ask, do you think that T-span 7 calcium channel interaction is druggable? And would it be of any use in terms of, controlling glucose levels in humans?
Yeah, so first of all, to the best of my knowledge, there's no drugs that affect the activity of T-span 7. But as far as a concept, I think it is a really cool concept, because there are, like Arya was talking about with the gamma subunits, there in that same family, there are subunits that interact with AMP receptors called TARPs. And so those, I know that people are drugging those AMP receptors with specific TARPs, like TARP accessory proteins. And so I think it would be possible in the future to perhaps target the calcium channels with T-span 7 as an accessory subunit.
And the drugs are
David said, I'm not sure if it would be good to reduce or over stimulate that interaction at this point, but
it should be druggable based on the TARP-based pharmacology that they have right now. And they're just looking, and for us, it would be really easy to do a drug screen because you could just overexpress both and see at what point you increased calcium channel activity, and then prove it was through TSPAN7 interaction or lack of interaction through the drug mediated. But the drugs are all just mediating the interactions of these protein complexes.
Thank you very much. It's very exciting and interesting, but we're running out of time now. I've got like a couple of more questions, but I guess I will have to keep them for later. I would like to thank everyone for attending and all the panelists for their time today. And I just want to remind everyone that there is a networking session starting pretty much in the next couple of minutes. So Rosie is going to post the link on the chat. So yeah, here it is. So everyone is more than welcome to join, and I hope I will see you in the next room in the next couple of minutes. And everyone have a nice day and stay safe.
thanks.
Thank you.